Okay, so today we're going to talk about DNA, RNA, and what we can make from them, how to make them, and everything. So, we mentioned earlier in the introduction that DNA and RNA is a little bit of an unusual place to start a biochemistry class. But the reason that we're doing this way is that it allows us to kind of set the ground rules, if you will, for how we're going to do things. And what I mean by that is things like, for example, how deeply we're going to get into structure. Structure is super, super important. Function follows structure. But some of you who have taken organic chemistry, say, in the past, have spent a lot of time drawing structures from a name of a molecule that the name took up, the whole line of a page, or naming some big cumbersome molecule that you were counting carbons out the ears, things like that. We're not going to do anything like that. What we are going to do, though, is point out the pertinent parts of the structure that give us the function we're talking about. Also, we're going to spend a lot of time doing a lot of processes. So in DNA, we get the opportunity to lay the groundwork both for how we're going to approach them from a teaching point of view, but how you're going to approach them from a learning point of view. My recommendation would be turn them into a story and be able to tell yourself the story. To me, that's more of an understanding approach. So we'll do some processes, smaller ones or less detailed ones, so you get to determine what's going to work for you there. Okay, with everything we do, we're going to first talk about what's the point. What's the point of the concept, the process? And what I mean by that is, why are we doing it? What's it have to do with biochemistry, with practice, things like that? So with DNA and RNA, our purpose is our cells need to know what kind of cell to be and how to be that kind of cell. They need to be able to use the information to do that. So we have DNA, we have RNA, we're ultimately going to have proteins for that purpose. We're going to use a decent number of analogies in our discussions, and the first one is for our genetic code. So let's think first about our everyday language. In our everyday language, we have letters. We put the letters together to form a word. The letters on their own don't have any meaning, really. The word has some meaning, but to really convey a piece of information, we need to put those words together, and that forms the thought, forms the sentence. The sentence is the piece of information that we're trying to convey. So in terms of our genetic code, we have nucleotides, and we code those nucleotides with letters, A, T, C, so on. And we'll talk about that in a minute. C for cytosine, A for adenine. We put 3 letters together to code for an amino acid. The amino acid is like the word. Then, as you probably already know, proteins are made up of amino acids. We put the protein together by connecting, and we'll talk about how, we'll talk about the bonding, by connecting the individual amino acids. That protein is the piece of information that the cell uses, has, and can act on or convey. So ultimately where we're going with this is we are going to make proteins. So a couple things first, though, before we get there. Some of you who have taken biochem before are looking at this and saying, oh no, do I have to memorize that again? And my answer is no. at least not for now, and we might come back to it. But what you do need to know for now, know how to code for stop and know how to code for start. The rest of it we'll talk about more, but for now you don't have to memorize it. Okay, there's some things about our genetic code. Our genetic code is universal. Your genetic code is my genetic code and everybody else's genetic code. Unambiguous. When we're coding for leucine, we're coding for leucine. Redundant. Like our everyday language, we have synonyms. We have multiple words that have the same meaning. We have multiple ways of coding for the same amino acid. It's non-overlapping and commaless. That's a little different than our everyday language. So let's think about language. When we have a sentence, we have some indicator at the beginning of a sentence saying we're starting a new sentence now. We capitalize the first letter of the first word. Then we have a word, a space, a word, a space, word, space. Then finally, some punctuation on the end. So what are we doing there? With the capital letter, we're indicating we're starting here. But when we're coding for a protein, putting amino acids together, remember we can code for start. So we code for start. Then we have a word space, word space, word space. We have to do that because otherwise we don't know where one word ends and another one starts. But with our genetic code, each amino acid is 3 letters. So we know it's 3333. Then in our sentence, we have punctuation of some form on the end to say, we're done now. This thought is over. I'm going to put a space and start a new one. We have a way of coding for stop. So we have stop. So that's our non-overlapping and commaless. So our genetic code consists of bases, and we have 4 bases: adenine, thymine, cytosine, and guanine. Be a little careful with this. We have thymine. Don't confuse it with vitamin B1. Remember, vitamin B1 is thiamine. So keep in mind, all of your testing, including the class and national boards, is going to be multiple choice. And I know at least when I first came into a multiple choice world, I thought, how easy is this? The answer is right in front of you. But the answer is right in front of you. But there's 3 other things are right in front of you called distractors. That's probably the most appropriate term you could possibly have for the wrong answers, because the more you stare at them, the more they distract you. So the devil's in the details with multiple choice. One little thing can take a right answer and make it a wrong answer, or vice versa. So little details like this, can make or break you. So anyway, we have 4 bases in combinations, possible combinations of 3, for 64 possible codons. Okay, so the DNA molecule is a double helix. I know you've heard that over and over and over. Rosalind Franklin is the one that figured that out. And it's made up of nucleotides. The nucleotides contain adenine, cytosine, thymine, or guanine. There's equal amounts of both. 2 of those bases are considered purines. We'll come back and we'll talk about that structure. We'll do a little short video sometime and draw them. 2 of them are considered pyrimidines. And again, we'll look at the structure in a little short video. You do have to know which is which. A goofy little mnemonic for the purines is, I can't topple the pyramids. C for cytosine, T for thymine, pyramids for pyrimidines. So Watson and Crick proposed that DNA is a double-stranded helix with anti-parallel strands and bases linked by hydrogen bonds. So we'll talk about that too. Okay, so let's talk about structure. And this is where, like I said, we're kind of laying the groundwork a little bit for structure. First, though, let's look at the building block of a DNA molecule. So let's look at the bottom corner here. What we have is we have a sugar, and we should probably actually have an oxygen showing at the very top of it. Let's try that again. So we have the oxygen here. Then, as we go around, we have carbon 1, 2, 3, 4, and carbon 5 outside the ring. So this is our building block of a DNA molecule. We have our base attached to carbon 1, and on carbon 5, we have 3 phosphates attached. Those phosphates is something we're going to talk about more later, and we're going to talk about them quite a bit in a couple of contexts. And this is the reason we're taking the time now to talk about this building block. These phosphates are attached in high energy phosphate bonds. So when we see these molecules that we'll abbreviate, most often the one we're going to see is ATP. The base is adenine. We put that together with the sugar, get adenosine, TP for triphosphate. So most often it's going to be adenosine triphosphate, and we'll make it and we'll use it and we'll make it and we'll use it. It's an energy molecule. So what I mean by that is we're going to use the energy contained in these high energy bonds. So what we'll do is, let's say we need energy for something. We'll break a high energy phosphate bond. We'll definitely use the energy for something. We might or might not use the phosphate. If we need more energy, we might break another high energy phosphate bond. Again, might use the phosphate or we might not. Then we'll eat something. So we'll take in food, take in fuel. We'll go through all the steps, reactions, processes, and everything that we haven't done yet. We'll replace the energy in a high energy phosphate bond and connect to phosphate. Replace the energy in another one, connect to phosphate. Most often we just take this phosphate on and off. It's more efficient that way. But what you're going to start doing as we start moving through our topics and everything is you're going to start keeping a really close eye on these phosphates, and more so the high energy phosphate bonds. It's kind of like a budgeting project. I'm going to have some reactions that are going to need energy, so I'll take phosphates off, break the high energy phosphate bonds, and get that energy to use for whatever I'm doing. I'm going to have some reactions that give me energy. I'll replace the energy in a high energy phosphate bond and attach a phosphate. So we're always going to be using ATP, we're always going to be making ATP. We'll use money as an analogy fairly frequently. A lot of you don't like my money analogy, so let me throw it out there right now. I know you're not in this profession. It's not about the money, it's about helping people. I get that. But in our everyday life, in your office, in most things, you've got to manage money. So in your office, you're always going to have money coming in. You're always going to be spending money. So you've always got to keep track of both, keep the balance, make sure you have more money coming in than you're spending. So when I use the analogies, it's tapping into that ability you already have in your everyday life. So with our ATP, we're always going to be making ATP. We're always going to be using ATP. We just have to make sure that we're making enough to have it available for when we need to use it. So let's go back. Okay, so let's look at our DNA molecule. So we have that sugar. On carbon 1, we have a base attached, in this case adenine. Carbon 2, nothing. Carbon 3 we'll talk about in a minute. Carbon 4, nothing. Carbon 5 has a phosphate. When we put this molecule in, or added it, it had 2 more phosphates. We took them off, broke the high energy phosphate bond, and used this phosphate. So what we used that phosphate for was to connect to the carbon 3 of another sugar. So again, we have the oxygen. We count clockwise from the oxygen. Carbon 1 has the base attached. 2, nothing. 3 is our attachment point to the phosphate. The phosphate that came with the other sugar. 4, nothing. 5 to its phosphate. Phosphate that came with that sugar, had 2 more attached, took those off. So that's where our structure comes from in terms of the side of the DNA molecule. If we look at the DNA molecule kind of like a big ladder, this is how we form the side. There's a term for that bond. We call that bond, get all this stuff out of the way. We call that bond a phosphodiester bond. So the term is phosphodiester. And I broke it up on purpose in pieces like that. So remember what an ester is. And we're going to come back and talk about functional groups also. So let's just do it. Let's talk about an ester. I have an ester, I have a carbon, double bonded to an oxygen, single bonded to an oxygen, and something attached to either side. In chemistry, we use an R as our variable, kind of like the X in math. So that's our ester. And what functional groups are is a name for an arrangement of a particular part of a molecule. And we're going to do a section on functional groups, and you'll learn them and have to know the names and everything. So what they really are is, rather than every time we want to describe this arrangement of a molecule, we'll say we have an ester. Rather than saying I have a carbon double bonded to one oxygen, single bonded to another oxygen, and both ends attached to something else. Kind of cumbersome to keep saying. We can just say ester. So if you look at this phosphate, we kind of have the ester idea. Change the carbon to a phosphorus. We have an ester here. We have an ester here. So that's our diester part. So carbon changed to a phosphorus is our phospho. 2 of them is our di and our ester. So that's how this becomes a phosphodiester bond. The phosphodiester bond is formed between carbon 5 and carbon 3. Carbon 5 of one sugar, carbon 3 of another sugar. So we have a phosphodiester bond attaching a 5 prime to a 3 prime, 5 prime to a 3 prime, and so on. So that's how we form the sides of the DNA molecule. But there's a couple more things that are interesting about the sides of the DNA molecule. If you look at the sides of our DNA molecule, you'll notice that if we want to call, let's look at this sugar, we have the oxygen on the top of the sugar, oxygen on the top, and so on. If you look at the other side, the oxygen's on the bottom. So here it is on the bottom of the sugar, bottom of the sugar. So what we call that is a parallel-anti-parallel structure. So it's kind of like if we want to call one side right side up, we're going to call the other side upside down. So that's our parallel-anti-parallel. structure of a DNA molecule. So with that parallel, anti-parallel structure, there's a couple other things that you're going to notice. The molecule, of course, ends, and if you look at what carbon is the final one with nothing attached to it on one side, it's carbon 3. So that's our 3' end. The other side is carbon 5. Phosphate doesn't count. It's the attacher. So we'll say things like we have the carbon 5 unattached. So the 5' end up here, got a carbon 5 unattached. Remember, the attacher doesn't count. 5' end, carbon 3 unattached. So our 3' end. So that's part of our parallel, anti-parallel structure, or I guess really I should say the consequence of our parallel, anti-parallel structure. So that's the sides of our ladder. Then we look at the middle. When we do this parallel, anti-parallel arrangement, that sticks our bases out into the middle. So what we do is we line up what base we're matching to what base. So we're always going to match a purine to a pyrimidine. So I got my pyrimidines, my thymine, my cytosine. I'm going to match them to particular purines. Thymine gets attached to adenine. Cytosine gets attached to guanine. So what I attach those with is hydrogen bonds. So I attach adenine to thymine with 2 hydrogen bonds. I attach cytosine to guanine with 3 hydrogen bonds. So everything I say is in the PowerPoint, so you can very easily read the PowerPoint. And I try to be very careful not to say anything that's not in the PowerPoint. This, though, may not be spelled out in the PowerPoint, the number of bonds. So make sure you know I have 3 hydrogen bonds attaching cytosine to guanine. I have 2 attaching adenine to thymine. Goofy way of remembering it, and I'll give you my goofy ways of remembering things. If you find them helpful, use them. If you don't. But at is a little 2-letter word. When I put adenine and thymine together, I get that little 2-letter word using 2 hydrogen bonds. Told you it was goofy. Okay, so that's our DNA structure. And next thing we're going to do is we're going to talk about using that. What are we going to do with it? So next thing we're going to do is we're going to do 3 processes. 3 fairly short processes. And what I mean by short isn't necessarily that they're short, but we only have to get into things to a certain depth in terms of the scope of this course. The scope of this course is for you to learn biochemistry to be a better chiropractor. We know going into this class exactly what you're doing. You want to be a really good chiropractor, taking really good care of your patients, really helping them. So that determines our depth on a lot of things. As opposed to an undergraduate class, if you were teaching that, and I don't know if you're going to chiro college, pharmacy school, dental, med, PhD in biochemistry. And that makes it much more difficult to determine how deeply to go into things. So in a way, this is really easier, I guess, both for you and for me with it. So anyway, we're going to do 3 processes, and it gives us a chance to lay the groundwork for how we're going to do processes, how we're going to approach processes. So the first process we're going to do is we're going to do DNA replication. That's where we take DNA that we have and we make more new DNA. So we said with everything that we do, you got to know the point of the process. So the point of replication is to make new DNA. Well, when do I need new DNA? Well, I need new DNA when I'm making new cells. I'm always making new cells. There'll be points in your life where you're making more new cells than other times. When you're a little baby, growing up, say, to be a big baby or a little kid or whatever. Pregnant moms, maybe pathologies like a cancer growing or something. So there'll be times that you're always making new cells, but there'll be times you're making more than others. So we'll do that. We'll make DNA out of DNA that we already have. Another thing you need to know is where we're going to do these processes. So with DNA, we're going to make more DNA in the nucleus. And what you're going to notice is that when we have the where question, you have to know where we're doing stuff, making stuff, on two levels. Have to know on the cellular level and have to know on the tissue level. So cellular level is nucleus, tissue level is all tissues. So not anything too exciting there. As we get going, you're going to notice some real trends in the wheres on stuff. And we're going to take advantage of those trends. So, and that's where our application comes in too. So when we get going on metabolism, you're going to notice things like we do a lot of our storage processes, store extra carbohydrates, store fats. Storage stuff is usually the cytoplasm. You're going to notice a lot of our energy making stuff in the mitochondria. We're going to totally use that trend. Then you're going to notice things on the tissue level that you can use too. Like the muscle will always be about doing or not doing. You're exercising, you're laying on the couch, things like that. And that'll have a lot to do with what's going on in the muscle. The liver is a really, really interesting organ. The liver, we'll say things like the liver's like everybody's mama, cleans up after everybody, takes care of everybody. But we say that because it's really pretty much true. Liver feeds everybody, meaning all of your tissues. So the liver worries about your blood glucose levels. Well, where's the blood go? All of your tissues. So we'll use that. The other thing you'll notice with the liver is if we have some molecule that you don't want, what do we do with it? We send it back to the liver. The liver cleans up after everybody. So again, these little processes, as I keep calling them, are kind of setting the stage, setting the groundwork for the time when we get into more major in terms of our coverage, the depth to which we go kind of processes. So we'll make DNA out of DNA that we have, and we do that when we're making new cells. But here's the thing with DNA. It's kind of all wrapped up in the nucleus and not really doing a lot. So let's say you just got a really good deal on a brand new biochemistry book. It was like 75% off. So you got that brand new chemistry book, and I'll tell you in a minute why you got such a good deal on it. You got that brand new biochem book, and it came all wrapped up in cellophane. So that's kind of your nucleus, that's cellophane. And the DNA's in there, all wrapped up, not super accessible. So with your book, you got to rip that cellophane off so you can get into the book and get your information. With our DNA, we've got to get that information out to the cell. The way we're going to do that is we're going to do a process called transcription that's going to make a message, as in messenger RNA, that we can send out into the cell. So the DNA acts as the pattern, contains the information that we want to use to make messenger RNA to send out into the cell. So we're going to do it in the nucleus, and again, in all tissues. So you rip the cellophane off, got the information, open the book up, you're good to go. Well, kind of. So you thought. Here's why you got such a good deal on the book. We send that messenger RNA out into the cell, and the cell says, well, great, thanks. I don't speak RNA. You open your book up and it's written in Greek. Oh, great. Now I know why I got the $100 book for $20. I don't speak Greek. So what do I need to do? We need to translate that. So the cell is saying, great, thanks a lot there, Nucleus. You sent me the message. It's in Greek. I don't speak Greek. We need to translate it. So what we're going to do is we're going to take the information contained in that messenger RNA. We're going to make a protein. And the cell is happy now. The cell speaks protein. We're all good. Has the information it needs. You got somebody, your Greek friends say, to translate that book into English. Good to go. So we're going to do this on the ribosome. And again, all tissues. So those are the three processes we're going to do. When we get really rolling, next try, and we're doing tons and tons of processes, you're going to start saying, okay, what do I have to know? Need to know what the point of the process is. What am I doing? What am I making? Where am I doing it? Cellular level? Where am I doing it? Tissue level. Okay, so let's get into our first process. So our first process is DNA replication. We have some hypotheses on how we do that, and we do it, what most researchers agree on, is we do it in a semi-conservative manner. So we do it in a semi-conservative manner. What I mean by that is we open up the DNA strand, we copy half, and we copy half, as opposed to a conservative manner where we plunk it on the photocopier and copy it, or a dispersive manner where we randomly just copy pieces here and there. So we copy DNA in a semi-conservative manner. So when we're building this strand, we're going to build the strand by adding this building block we were talking about a few minutes ago. So this building block that we have, the nucleotide, we have the sugar. On carbon 1 we have a base. On carbon 5 we have 3 phosphates. We're going to break off 2 out of those 3 phosphates. So we'll take those 2 phosphates off. We're not going to do anything with them. Couple things with that. One is, when we break them off, we're breaking high energy phosphate bonds to get those phosphates. We're all about the energy. That's what we want. The other thing is, within the scope of this class, there's going to be certain molecules and parts of molecules that we're going to assume if we don't need them, we're just going to leave them there. Or if we need this particular molecule or part of a molecule, we're going to assume it's hanging out there waiting for us. Not completely true on both of those things, but it's a scope thing. Other classes we'll talk about, usually physiology, what you do with the water we just left hanging out there, or what you do with the carbon dioxide we just left hanging out there, or how is that water hanging out there waiting for you, or maybe your nutrition class will talk about what you ate that phosphate's there. So like any academic program, there's always a scope where this class goes up to the line here, and here's where another class takes over. Of course, between biochem and physiology, it's kind of a big wide gray line. So you'll see us kind of crossing back and forth on that here and there. So that's the building block we're going to add on. So let's talk about enzymes for a minute. And we're going to tell most of our story on a picture, but let's talk about enzymes. So we're going to talk about enzymes in terms of what they do, how they do it, and all that. We'll also talk about naming them. We'll do a separate section on that, either next module or the one after. I think it's next. So enzyme names very casually are what they do or what they do it to with an A's on the end. When you see a molecule name ending in A's, that's an enzyme. So one of the things we're going to do as we go through this is we're going to build up your biochem terminology list, I guess, by you, for you. So I'd probably put a little document somewhere with some of this stuff because it will get really important later when you're trying to figure out questions and terminology and things like that. So if it ends in A's, it's an enzyme. So we're going to go to the picture and we'll tell the story. All the enzymes that we're going to talk about and use are here on this page. So it's always easier to tell a story with a picture, and that's what we'll do. So what we're going to do is we're going to talk about replication. So I want to replicate my DNA molecule. What I really mean by that is I want to use the DNA that I have as a pattern to make more new DNA. So, not a great picture, but better than what I can draw. So what we have is we have what we call the parent DNA molecule. We know that we want to unwind that. So these pieces here are our DNA molecule, the blue part, is the parent DNA molecule unwound. The little teeth sticking out, those are our bases. So we have, say adenine, guanine, whatever, sticking out. So they're sticking out, and we're going to match those with new bases as we're growing our new DNA molecule. So when we see the little teeth of the two combs meeting, that's our matches. So in this diagram, we don't have them labeled, just know that we're matching as we do it. We're putting an adenine with a thymine or a cytosine with a guanine. So when we draw or when we make this molecule, we're going to grow a DNA molecule in a 5' to a 3' direction. So what we're going to do is we're going to unwind it. So we're going to unwind it with an enzyme called a helicase. Kind of descriptive. We're unwinding the helix with a helicase. That gives us, exposes to us, the two sides of the DNA molecule. So what we want to do is we want to grow this molecule in a 5' to a 3' direction. So we're going to build it on this side, this way. Go to 3'. So we have a 5'. We can grow it that way. So what we're going to do is keep adding our matching base. Add our cytosine to our guanine, our adenine to our thymine to an adenine as we go along. And we'll grow the molecule. The enzyme that's doing that, as in elongates, is DNA polymerase 3. DNA polymerase 3 attaches a 5 to a 3. So we're building the molecule. But on the other strand, we have a problem. We can't go that direction. We can build in a 5 to 3. We're reading 3 to 5, and we don't have a 3'. We have a 5'. So what we're going to do this time is we've got to build it kind of in pieces. So what we're going to do is we're going to build in little segments. So we'll build in little segments. So the way we're going to do that is we've got to get in there, find our spot to get going, start the process. And when we do that on both strands, actually, and I forgot to say it on the other strand, we have a DNA, actually, totally messing up. We have a primer. Go in and do that. and it's actually a piece of RNA. So it's an RNA primer primase. It goes in and gets us our spot to start, does it on both strands. But what we've got to do is we've got to dig in, put our primer in there, make our strand. Dig in, make our strand. Dig in, make our strand. And what's different with this is we're kind of going upstream a little bit, if you will, on this one. So it's like swimming up a river. You're going to swim a little ways, run out of energy, stop, swim again, run out of energy, stop. So we're going to do little fragments. We'll go do a little piece, do a little piece. So we have to put the primer in each time to do that. So we're going to make the DNA molecule in pieces. So what we need to do, make the fragments, and we do. Then we've got to get the primer out of there. DNA polymerase 1 will undo the number 1 thing that you did. Get the primer out of there. Then we can link these fragments. Link these fragments with a ligase, and we'll now have a continuous piece of DNA. So this strand we were able to build this direction, this strand we're going to build this direction, but we're going to build it in pieces going upstream as we do it, ending up getting 2 new pieces of DNA from it. Okay, so a couple things, and a little bit of disclaimer in a way when we talk about this. We're going to talk about everything like it's a perfect process. Everything goes beautifully. We have beautiful DNA that we copy perfectly and exactly and make brand new perfect DNA. We don't. In this class, we're going to talk a lot about everything perfectly. You know, it's a normal biochemistry class. You'll get to things not going normally and perfectly later, and that's a class called pathology. But for now, we'll talk about everything like it does go perfectly. Recognizing, mentioning here and there, I guess probably more than recognizing, that it doesn't always, and we have mechanisms for repairing, fixing things that don't go perfectly. So one of those is a DNA proofreading mechanism that cuts our errors in replication down by a factor of about 1,000. Okay, a term that's probably come into the news more lately in the last few years is polymerase chain reactions. We haven't in the past spent an awful lot of time on it, mentioning a little more now because of the COVID testing. But what we're doing in a polymerase chain reaction is we're taking a DNA signal, we're replicating it in a test tube to amplify the signal so we can actually tell what that signal is, what that molecule is made up of. Okay, so next process we're going to do is transcription. A little bit of a shorter story here. But what we're going to do is we're going to make RNA from DNA that we already have. So there's a couple things we need to talk about with RNA. So you've probably known this since high school, but RNA is single-stranded. The sugar is a ribose rather than a deoxyribose. And rather than thymine, we have uracil as the 4th base. But let's talk for a second about this. I know you know it's single-stranded, it's ribose, it's uracil, but questions that I know you know have different statistical outcomes depending on how the question's written. If I ask you what base is in RNA instead of DNA, it'll be like 98, 99% stats, and always has been. If I ask you what base is in DNA rather than RNA, the stats will drop. Well, it's the same information regardless of which way I ask you. So what that's telling me is you're probably not reading the question, especially since 12 to 15% will pick uracil on that, even though it's asking what's in DNA. Thymine is in DNA. Back color for that. Thymine is in DNA. The uracil is in RNA. So with your questions, half the battle is reading the question, slowing down a little bit, reading the question, and making sure you're answering what's actually being asked. And I know sometimes that's a little challenging on a timed test, which they all are, but when you think about it, with any of our timed tests, what you're going to notice, there's going to be a lot of people out of there really, really quickly. You have time. So my point is slow down, read the question carefully, make sure you're answering what you are getting at or what you are being asked. All right, so we have different types of RNA. First one is messenger RNA. That's what we're going to make. It's the message. So it's the information from the DNA contained in a message that we're going to send out into the cell and make a protein from it. We're going to have tRNA or transfer RNA. That's going to act in translation, carry the specific amino acid to the growing polypeptide chain to make the protein. We're going to have ribosomal RNA. Kind of just hangs out in the ribosome. Well, kind of. It serves some purposes there. One of them is specificity. We're going to have a lot of stories about specificity and binding sites, what will match, what we need, what won't match, things like that. So the ribosomal RNA plays a role in the specificity, as in what amino acid do we need in what order, where, things like that. Okay, so what's a good place to draw this one? What we'll do, we'll actually use this slide as a blank slide and we'll just draw it. Everything's in here in words. It's just easier to draw it and talk our way through it. So what we're going to start out with is we have a piece of DNA. So we have a piece of DNA, and of course it has its bases. So I'll use the little comb teeth thing for that. And the first thing we've got to do is we've got to unzip that. So it's a nice long, long strand. And what we're going to do is we're going to kind of separate it in the middle and unzip it. So we have access to the bases because we're kind of using those as a pattern. So we're going to come in, unzip it so we can use it. So what we have when we have it unzipped is we have that DNA separated. We're only going to use one side of it. So we're going to use one side of it and make our messenger RNA. We're going to transcribe on one side. So we have a recognition sequence that says, get going. We're ready to make our new messenger RNA based on this DNA that we're using as a pattern. So what we'll do is we're going to have the complementary nucleotide float in. So if I have, I have a cytosine here. I'm going to have a guanine come in. I have an adenine. I'm going to have a uracil. Remember, uracil, not thymine, comes in, and so on. So my complementary nucleotides kind of float in there, forming us a chain. So remember, we had start, and then we have stop. So we keep going, and we make this thing. So we make this messenger RNA with these complementary nucleotides. form that chain, and it'll float away, allowing the DNA then to go back together. We can put the DNA back together, it goes and does its thing. But there's one little detail with this. What we actually made is what we call pre-RNA. Oh, and I almost forgot. We're adding all these nucleotides, growing the chain with RNA polymerase 2. So what am I making? I'm making RNA in the second process that we're talking about. So I get stop, and I have my pre-messenger RNA. What do I mean by pre? Well, when we make it, we have good parts and bad parts. So we have things called introns, and we have things called exons. So introns are non-coding parts. So they're in there, they're kind of dead weight in a way. Exons are the parts we want. They're the coding parts, they're the parts we want. So in that pre-RNA, we'll go back, we'll cut out the introns, put the exons back together. That's our messenger RNA that will go and carry the information out into the cell. So that's the part we really want. So kind of what we already said. Okay, we initiate it, we elongate it, we terminate it. Got the pre-mRNA, got the introns, the dead weight out of the way, sent the rest out into the cell. Okay, so next process we're going to do is translation. We sent that messenger RNA out into the cell. The cell said, thanks a lot, that's great, but I speak protein. So we've got to take the information contained in that messenger RNA, translate it, convert it into a protein, form a protein based on that information is the best way to say it. So what we're actually doing is we're coding for amino acids. So when we code for an amino acid, we're going to start with start. So remember, we have a word for start. Then we have the 3 nucleotides that code for each amino acid. So this is where, in our analogy, we're putting the sentence together. So the sentence is start, amino acid, amino acid, amino acid, however many, and stop, coding for it using our genetic code, our language as we defined. Okay, before we can do that, we need some stuff. So we need a couple of components. One of the components that we need is we need transfer RNA to carry our amino acids. So with our transfer RNA carrying our amino acids, we can start telling our specificity story. So the specificity is going to play a role in everything that we do. So we have a transfer RNA, which is some RNA kind of held together with hydrogen bonds, kind of actually in a little bit of a T shape. And what its job is to carry an amino acid up to the growing polypeptide chain. But it's specific. So a specific transfer RNA carries a specific amino acid. So it's not like I can take any transfer RNA I happen to have around and just grab onto an amino acid and transfer it. So I need the specific transfer RNA for the specific amino acid that I'm going to transfer. Then I'm going to put that transfer RNA and its specific amino acid together. I need an enzyme to do that. With enzymes, sometimes we're going to talk about enzymes individually, like this individual enzyme converts this individual substrate to this individual product. Or sometimes we're going to talk about enzymes in the sense that this family of enzymes does this to this family of molecules. Each individual member of the family is specific to an individual member of the enzyme family. So, amino acyl transfer RNA synthetases are a family of enzymes that connect specific transfer RNAs to specific amino acids. So we'll have a particular amino acyl transferase synthetase that's specific to a particular amino acid and its particular transfer RNA. Where we get the specificity is the binding site. Only that amino acid fits in that amino acid binding site. A different amino acid wouldn't fit. Only that specific transfer RNA fits in its binding site. It's kind of like, let's think of all of our front doors. We all have a front door with a lock on it, and we all have a key. But I can't take my key and go and open your door. I have a key, but it's not the one that fits your lock. I might have a transfer RNA, but I've got to have the right one to fit this enzyme. I have an amino acid, it's got to be the right one. So that's our specificity. My key is specific to my lock, your key is specific to your lock. I also have an ATP site, so that's telling me I need energy to do this. I'm going to use an ATP, spend an ATP, break that high energy phosphate bond because I need energy to do this. So what that amino acyl transfer RNA will do is put together that particular transfer RNA with its particular amino acid, use the ATP to do it, and make what we call a charged transfer RNA. So then what we're going to do is we're going to take this story to the ribosome. So our little blobs on here are a ribosome, and our ribosome has a large subunit and a small subunit. So I have my small subunit, I have my large subunit. Same thing here, small subunit and large subunit. The little comb looking thing that we have going through here, that's my messenger RNA. So that's something you could look at as kind of a list. That's telling me I want this amino acid, then this amino acid, and this amino acid. So an analogy we use for this sometimes is that of we're gonna start our own Instacart business. We'll talk about that in a minute. So first, before we do that, let's talk about the sites on the large subunit. And they're described on another slide, so I'm not going to say anything that's not also in the PowerPoint. But we have 4 sites. Some books only talk about 3, but I'd rather you know more stuff than you need than less stuff than you need. So the T site is a transfer site. It's kind of like when you're playing baseball when you're on deck. So your team just got up to bat, you're on deck, you're standing there stretching, swinging the bat, whatever. You're there, you're ready to go, but you're not really doing anything productive for the game in a sense. That's the T site. The transfer RNA holding its amino acid is on that site, ready to go. The next site is the A site. The A site is the amino acid site. That's where we match the amino acid to the messenger RNA. We'll say, look, messenger RNA, you wanted leucine? Here you go. I got your leucine. You wanted phenylalanine? I got your phenylalanine, whatever. So the transfer RNA went from the T site to the A site. After we do the match, we're going to come over to the P site. The P site is the polypeptide site. So a protein is really just a chain of amino acids attached with a peptide bond, and we'll come back and talk about the bonding and the arrangement and everything of that. So I've got to join the amino acids in this growing polypeptide chain. That's what happens on the P site. So in effect, it's a little bit of a drop-off. The transfer RNA drops off the amino acid, attaches it to the growing polypeptide chain. Then it will move over to the E site, exits. So that's where the transfer RNA has done its job, dropped off its amino acid, goes to the E site, the exit site, and exits. So now let's talk about our business. We're going to go into business and we're going to put Instacart out of business. We can do a much better job, a much more efficient job. So here's how it's going to work. It's going to be kind of like translation. So I'm going to be messenger RNA in our business. And what that means is I have the list. I have the grocery list. So it's like messenger RNA says start. They'll say I want alanine, I want tyrosine, I want leucine, whatever. It's calling out a list. So then all of you guys in the class are going to be transfer RNA. So just like transfer RNA, you're going to be specific to your grocery item, whatever it is. You might be ketchup, mustard, hot dog, I don't know, milk, bread, whatever. So whatever you're specific to, that's what you carry. So what you're going to do is you're going to get your grocery item, whatever it is. You'll get your ketchup, you'll get your mustard, you'll get whatever. I'm going to be going through the list, calling it out. So let's say you're ketchup. You know you're next. So you're hanging out on the T side, holding on to your ketchup. Then everybody moves over one. You're going to come over then to the A side. Say, look, messenger RNA, you want a ketchup. You asked for it. Here you go. I got you a ketchup. So now we're matching. Then you're going to come over to the P side. You're going to throw your ketchup into the shopping cart. Get rid of it. Throw it into the shopping cart. You dropped it off. You delivered it. You'll go to the E side, exit, go get another bottle of ketchup. While mustard's behind you doing the same thing. Want mustard? Here you go. Got your mustard. Drop off the mustard. Come to the E side, exit, and so on. You'll move down the chain that way. Like we're building up our list, throwing all the groceries that we wanted based on that list in the shopping cart. We're building up that polypeptide chain by joining an amino acid to an amino acid to an amino acid. And again, remember we said we code for stop. We get to stop. That's the end of our amino acid. We have our protein. We've translated. We're done. We have what we wanted. So another picture of the same thing that we talked through. Names of the sites that we talked to. We're going to grow, and we'll talk about this some more when we talk about amino acid structure. We're going to grow from the N-terminus, the N that contains the nitrogen, to the C-terminus, the N that contains the carbon. So we'll get there when we talk about amino acid structure, talk about where the nitrogen is as part of the amino group, where the carbon is as part of the carboxylic acid. So we get to our stop signal and we stop, messenger RNA goes on its way. The other thing we're good at doing, and this is just kind of in the way we think, we have one-track minds, and that affects us in the learning process. We think about one thing at a time because we can learn one thing at a time. What that leads us to do, though, is kind of think like and act like we have one thing going on in the cell, in the tissue, in the organism at a time. One of the aha moments of this class is when you realize, no, we have lots of different stuff going on all at the same time in lots of different places. The real aha moment of it is when you remember that I might have two processes that have opposite functions going on at the same time, just in two different places. And we'll talk about that. We'll kind of bring you back to that when you have stuff going on, but you can't quite put the pieces together. So with that in mind, we're going to talking about we have one messenger RNA and one ribosome making one protein, and that's it. And that's not true at all. We could have one messenger RNA interacting with a whole bunch of ribosomes, making a whole bunch of proteins all at once. The other thing we're really good at doing is talking about a process and acting like we got to the end, put a period on it, we're done. And as far as that process is defined, yeah, we might be done, but we're never done. So what I mean by that is we got the protein, so translation's done, mission accomplished, put a period on it, but the protein's not done. The protein might leave the cytoplasm, go into another organelle. It might go back to the nucleus. It might leave the cell altogether. We also act like that protein is done. It's going to stay the way it is forever and ever. That's not the case necessarily either. We might break it up. Proteolysis. Lysis is breaking. We might add glucoses to it. We'll talk about when and why we do that. That's not always a good thing. We might add phosphates to it. So again, we're never done. Let's talk about regulation. And before I bring up the regulation or put it on the side, let's just talk about it for a minute. So here's the thing with regulation. We need a way of having our cells do what we need them to do and not do what we don't need them to do. And what I mean by that is we need a way of having our cells make us the stuff we need and not make us the stuff that we don't need because we already have it. The way we do that is through regulation. So let's say, well, let's do this one. Let's say I have a molecule of glucose. And glucose is a molecule that we're going to spend a lot of time with. It can go two different directions. It can go to energy, or it can go to storage. So let's say I just got up, I haven't eaten since last night, I just got up first thing in the morning, and I ate a glucose. So what do I want to do? What I want to do is I want to send it to energy and not to storage. So that's regulation. I shut down this path so it can go to energy. Then let's say I keep eating. I keep eating more glucoses. Well, what do I want to do? What I want to do is now energy's taken care of. I want to shut down that path and I want to open up this one and save the rest of that glucose for later. So that's regulation. So let's take it one step further. Let's say now I have a process. And I have enzymes catalyzing each step of my process. So let's say I want to regulate, and the regulation that I want to do is I want to prevent the formation of D. So I have A, and I want to prevent the formation of D. So we're talking about regulation like this to make your life easy. There's two ways of learning regulation. You can memorize it, and sometimes that's the quick answer. You know, you have a test tomorrow, you need to put out the immediate fire to get through the test. But here's your problem with memorization. As you know, it doesn't stay with you. You know, you memorize, regurgitate it for the test, and you probably don't know it the next day. Here's your really big problem with that, and it's twofold. One is, you're going to know this stuff for your subsequent courses to learn how to take really good care of your patients. But from a more immediate perspective, you're going to take part one of the national boards in about a year. And during that year, you're not going to directly be doing biochem. You'll be using it in other classes, but not directly doing it. So there's a really good opportunity to forget a lot of it. The other thing with that year is you're going to be learning a lot of other stuff. So Part 1 of the national boards is really inconvenient. It's not a difficult test, and I do talk about it a lot because I want you prepared for it, but I don't want you afraid of it. That's not why I talk about it. I want you prepared. And if there's something I think of that might be helpful to you, I'll tell you while I think of it because I might not think of it later. So it's not a difficult test, it's an inconvenient test. One of the inconveniences is you're learning your biochem now and going to take Part 1 in trimester 4, like a year from now. So you've just had a ton of time to forget it. So it'd be kind of like getting ready now for a test on the first 2 years of your undergrad. You did it, you learned it, you knew it, you did well in it, or you wouldn't be here right now, but you forgot a lot of it. And you're busy this try, so it wouldn't be convenient to have to relearn it again now. So it's that kind of thing. So the reason I'm telling you this is putting something in the understanding category takes more time and effort. And I know you're going to have times during this term that you're not going to feel like you have the time. Make the time and do it, because you have more time now than you will the trimester before boards. And what you get in the understanding category in your head will stay with you. What's in the memorizing category, you're just going to have to do it over again. So do regulation from the understanding approach. So if my goal is to prevent the formation of D, I could do a number of different things. I can shut down, say, enzyme 1 and prevent the formation of B. Or I could leave 1 alone, form B, then get rid of it. Then it's not there to make C or D. Or I could make B, leave it alone, shut down enzyme 2. That keeps me from making C, which will keep me from making D. Or leave B alone, leave enzyme 2 alone, make C and destroy it. Or leave C alone, shut down enzyme 3, and so on. If I do anything, whether it be destroy a product so it's not there to be the next reactant, or shut down an enzyme so it's not there to make the product that will become the next reactant, do either of those two things in any of these steps, I'm going to prevent the formation of D. There's regulation done. So let's look at all the junk. And I'm saying junk on purpose, because it's work to memorize stuff you don't need to memorize. So what it is a list of things I can do to regulate. So I can control the synthesis activity by regulating or controlling the production of enzymes. I can get rid of one of the enzymes that would make a product that without the enzyme I don't get the product, so the product's not there to be the next reactant. I can block transcription. If I don't do transcription, I don't get messenger RNA. If I don't get messenger RNA, I can't make a protein. Mission accomplished. Or I could take the messenger RNA and hydrolyze it, break it up after it's made. Again, no messenger RNA. I can't put it with a ribosome to do translation and make a protein, so no protein. I can prevent translation of the messenger RNA at the ribosome. Don't let the messenger RNA interact with the ribosome. I'm not going to get a protein. Or, final one, I could make the protein and destroy it. If I destroy it, I don't have the protein to go and do whatever that protein was going to do. Okay, let's talk about pharmaceuticals. So, a couple things with pharmaceuticals. One is, if you ever want to pick a fight in a room full of chiropractors, express some strong opinions about whether or not chiropractors should prescribe. You'll get some very strong opinions there. With that, I try really, really hard, not always successfully, not But anyway, not always successfully, but I try really hard not to give you any of my opinions. And I think all faculty should do that because it's not your job to take our opinions. It's your job to form your own. So a couple of things with that. One is form your own opinions. Don't just take somebody else's. That's not too difficult. Here's the hard part. As you gather more information, which you will during this program and after this program, allow yourself to change your opinion. That's the hard thing, because we feel like when we change our opinion, we're saying we were wrong. You're not saying you were wrong. You're saying you gathered more information, and now, as a result of that information, you have a new opinion. So anyway, whether we should prescribe or not doesn't matter for this. Whether you believe we should prescribe or not, you need to pay really, really close attention in pharmacology class. And here's why. Whether you prescribe or not, your patients are going to have been prescribed to. So, regardless of your feelings on prescription rights for chiropractors, you need to know a lot about prescriptions because your patients will have been taking a lot of prescriptions. So you need to know how they act. So what you're going to do is the common ones you're going to get to know, and you'll know their mechanisms and everything pretty well. The ones that aren't as common, that's okay. Because what you're going to do is you're going to train your staff to give you the paperwork before they give you the patient. That way you can go over the paperwork and know what's going on with the patient before you're actually sitting in front of them having a conversation. So when you look at the list of medications they're on, if you need to look anything up, go look it up. Nothing wrong with that. And that's how you're going to get to know the common medications. So when you look them up, what you're going to find is a mechanism. What that mechanism is it's going to be some kind of regulation. And you'll look and you'll see, well, this is where I either inhibit an enzyme to keep from making a product, so that product's not there then to be reacted in the next step, or I take that product and I get rid of it so it's not there to be a reactant in the next step. Either way, not making that final end product that the goal of that pharmaceutical is to not make. So it's just going to be regulation. So here's an example of some common antibiotics. And like I said, don't worry about knowing them, but look at the mechanisms of them. All they are is steps, regulation, like what we talked about. First one, I don't form peptide bonds. I don't connect the amino acids to each other. I don't connect the amino acids, I don't get a protein. So I've regulated. Second one, I don't let the messenger RNA move along the ribosome. If it can't move, it can't call for the amino acids it wants to make the protein. No protein. I don't let the transfer RNA interact with the messenger RNA. Well, if the transfer RNA can't bring the amino acids up and make the protein, no protein. Fourth one, I don't let translation initiate. I don't start it. If I don't start it, I don't get a protein. What's the last one? I don't let the transfer RNA bind to the ribosome. The transfer RNA can't interact with the ribosome again. No protein. So again, mechanisms are nothing more than regulation in some form or another. Okay, last thing we're going to talk about is mutations. So we talked about building perfect DNA, making perfect messenger RNA to make our perfect proteins. Like we said, even though this is a normal biochemistry class, and we're going to act like everything happens perfectly and beautifully, we recognize that that's not necessarily the case. So we have some definitions that describe the errors that can happen. So the error might happen in the formation, might happen afterwards, things like that. And the error might be a single wrong, we'll say wrong letter, using the analogy of a language, or it might be chromosomal, wrong part of, say, a sentence, again, using our analogy. So there's different kinds of point mutations, wrong letter. Might be silent. We might just so happen to switch out a letter but code for the same amino acid. So silent makes no change, we get the same protein. Might be missense, like mistake, code for the wrong amino acid. Put the wrong word in a sentence, we change the meaning of the sentence. Could be nonsense, meaning we put stop in before we mean to put stop, like cutting off a sentence in the middle. You're not getting the complete protein, like you're not getting the complete piece of information. Might be a frame shift. We add an extra letter in there, shift everything over by one letter, remembering we're reading in sets of 3, change the whole thing completely. Then we can have chromosomal mutations. This is more than one letter being wrong in the sentence. We might delete part of it. So we delete part of it, we shift the whole reading frame with our 333. Might duplicate it. We duplicate it, we get multiple copies. We end up with an increased amount of the gene. Invert it, reverse the orientation, flip it over, change the meaning. Translocate it, move pieces around, changing the meaning. Okay, that's our coverage of DNA and RNA. So like we said, that's a little bit of an introduction, obviously, to the content, but also to our approach, how we're approaching structure, things like that, depth. So we'll stop with our DNA, RNA there. Next place we're going to go is water. And again, a little bit of an unusual order to do things with. Not as much with the water as it was with the DNA and the RNA, but it allows us to continue laying our groundwork for our approach, particularly in this case with bonding. So we'll do some questions, we'll do a quiz and everything with our DNA, then we'll move on to water.